Picture this. You're a biology student, first week of class, and your professor puts two photographs side by side on the projector. On the left, a dolphin. On the right, an ichthyosaur, a marine reptile that went extinct approximately 90 million years ago. The similarity is so immediate and so complete that several students assume there's been a mistake, that the professor accidentally uploaded the same image twice, or that one of these is a dolphin fossil and the other is a living dolphin. Same streamlined torpedo body, same dorsal fin, same fluked tail, same pointed snout, same general body proportions, same swimming posture, same profile in the water. One is a mammal, the other was a reptile. They share no common ancestor anywhere near the point where either of them developed those features. The mammal lineage and the reptile lineage had been separate for hundreds of millions of years before the ichthyosaur and the ancestors of dolphins independently arrived at, essentially, the same design. Your professor smiles at the class's confusion. Evolution, she says, has a limited imagination. She is, in the most illuminating way possible, correct. The phenomenon she's describing has a formal name, convergent evolution, and it is one of the most conceptually rich ideas in all of biology. It is the repeated, independent discovery of the same solutions to the same problems by organisms with completely different histories, separated by millions of years and vast phylogenetic distances. It is evolution doing the same thing twice, or 12 times, or 30 times, across wildly different starting points. And understanding why it happens reveals something profound about the nature of the biological world that goes far beyond, wow, those two animals look alike. The intellectual core of convergent evolution is the concept of adaptive landscapes. The idea that the space of possible biological forms is not uniformly accessible or uniformly viable, but is instead shaped by the physics and chemistry of the natural world into a terrain with peaks and valleys. The peaks are adaptive optima, body plans, structures, or physiological strategies that work extraordinarily well in specific environments. The valleys are the non-viable or inefficient forms between those peaks. Evolution, operating through natural selection, tends to move populations up the slopes of this landscape toward the peaks, because forms closer to the peak survive and reproduce more effectively than forms further away. Different populations, starting from different positions in the landscape, different evolutionary histories, different anatomical starting points, will nevertheless tend to converge on the same peaks if those peaks represent genuinely optimal solutions to the challenges of the environment. The dolphin and the ichthyosaur converged on the same body plan because that body plan, the hydrodynamic torpedo with stabilizing fins and a powerful tail, is a peak on the adaptive landscape for fast, active pursuit predation in open water. The physics of water resistance don't care whether you're a mammal or a reptile. A streamlined body reduces drag the same way, regardless of your ancestry. A crescent-shaped tail generates forward thrust through oscillation the same way, regardless of the molecular details of the muscles powering it. The peak is the peak, and evolution finds it from multiple directions because the landscape has the shape it has, determined by physical reality rather than biological history. This is the deep point. Convergent evolution is not a coincidence. It is not evolution being lazy or unimaginative. It is evidence that the space of viable biological solutions is constrained by the laws of physics, chemistry, and ecology in ways that make certain solutions not just good, but uniquely optimal. When we find the same form evolving multiple times independently, we are finding the fingerprints of those constraints, the signature of the adaptive peaks that the laws of the physical world create in biological space. The eye is the example that has generated the most philosophical discussion, and with good reason, because it is one of the most complex biological structures in existence, and it has evolved independently not once, not twice, but somewhere between 50 and 100 times depending on how you count and what you include in your definition of an eye. The vertebrate camera eye, the kind inside your head right now, and the cephalopod camera eye, the kind inside an octopus's head, are structurally almost identical. Both have a cornea, a lens that focuses light, a retina with photoreceptor cells, and an iris that regulates the amount of light entering. The similarity is extraordinary enough that it served as one of Darwin's most frustrating problems, The complexity of the eye was a favorite argument of those who doubted that natural selection could account for such elaborate structures, and here was the same elaborate structure appearing in two lineages that had been separated since before either had anything resembling a visual system. The crucial difference, the one that proves the independent origins, is in the wiring. In vertebrate eyes, the photoreceptor cells point away from the incoming light, with their connecting nerves running in front of the retina and bundling into the optic nerve through a gap that creates the blind spot. In cephalopod eyes, the photoreceptors point toward the incoming light, and the nerves run behind the retina, a more intuitively logical arrangement that doesn't create a blind spot. Same external design, different internal architecture, different evolutionary paths to the same functional outcome. The evolution of the eye from simple photosensitive patches involves perhaps 17 or so distinct evolutionary transitions, and the fact that lineages across the animal kingdom have made these transitions independently so many times is strong evidence that each step is not particularly improbable given the right starting conditions. The selective advantage of even the most primitive photosensitivity, being able to tell light from dark, is sufficient to initiate and maintain the evolutionary trajectory. And once started, the trajectory leads, via a series of individually advantageous steps, towards the same camera-style design that physics predicts should be optimal for resolving detailed images. Echolocation is a more cognitively demanding convergence than the eye, because it involves not just a structural feature, but a complete sensory system requiring coordinated evolution of sound production, acoustic anatomy, and neural processing specialized for interpreting returning echoes. Bats and toothed whales both echolocate. Both produce high-frequency sounds. Both have specialized inner ear structures tuned to those frequencies. Both have neural processing systems capable of extracting distance, direction, speed, and texture information from returning echoes. Both evolved these systems independently. The fossil record establishes that bat echolocation and cetacean echolocation developed at different times in different lineages from ancestors with no echolocation capacity. The convergence extends to the molecular level in ways that are particularly striking. When researchers compared the gene coding for Preston, a protein involved in the outer hair cells of the inner ear that amplifies sound detection, across echolocating bats, non-echolocating bats, toothed whales, and other mammals, they found that the Preston sequences in echolocating bats and toothed whales are more similar to each other than either is to their own closer relatives. The same mutations in the same protein, producing the same functional change, in two completely separate evolutionary events. This molecular convergence is more remarkable than morphological convergence because it involves the same specific molecular changes rather than the same general structural outcome. The space of possible Prestine mutations that improve echolocation sensitivity is apparently small enough that both lineages found the same solutions. The molecular adaptive landscape is as constrained as the morphological one. Shrews and tenrecs also show some degree of high-frequency hearing potentially related to echolocation-like abilities, representing additional independent origins of acoustic specialization in yet other lineages. The same physical principle, sound waves returning from objects encode spatial information that nervous systems can extract, is discovered repeatedly because it is genuinely discoverable and because the environments that favor it create strong selection pressure for any lineage that stumbles into the appropriate sensory territory. Wings are the convergence that most dramatically illustrates how the same functional requirement, powered flight, has been met by such radically different structural solutions that the convergence is architectural rather than detailed, and the differences reveal as much about evolutionary constraints as the similarities reveal about adaptive optima. Insects evolved flight approximately 320 million years ago, making them the first flying animals and the owners of the longest-running flight experiment in the history of life. Insect wings are outgrowths of the exoskeleton, flat extensions of the body wall that were elaborated into flight surfaces, powered by muscles that deform the thorax rather than attaching directly to the wing bases. Pterosaurs evolved flight independently approximately 230 million years ago, developing wings from the forelimbs by extending a single elongated fourth finger to support a membrane. Birds evolved flight from a different dinosaurian starting point, with wings built from the entire forelimb covered in feathers. A radically different structural solution than the pterosaur membrane. Bats evolved flight in the mammalian lineage somewhere around 50 to 60 million years ago, using a membrane stretched between elongated finger bones. Closer in structural approach to pterosaurs, but in completely different anatomy. Four completely independent origins of powered flight in animals, using four different structural solutions, all converging on the same functional requirement. By the way, if you want to go even deeper into the secret lives of ancient humans, I just launched a full course called The Secret Lives of Ancient Humans. 10 modules, 3+ hours of content covering everything we never talk about on YouTube. What they feared, how they communicated, what hunted them, and much more. Plus an exclusive PDF guide included. Link in the description. See you inside. Generating enough lift and thrust to overcome gravity and move through air. The physics of flight don't care about your ancestry. An airfoil generates lift through Bernoulli's principle regardless of what biological material it's made of. But the specific structural solutions differ because each lineage was working with different raw material. Exoskeleton, membrane over single digit, feather-covered forelimb, membrane over multiple digits. The peak on the adaptive landscape is flight, and multiple paths lead to that peak from different starting elevations in different parts of the morphological terrain. Venoms represent one of the most prolific examples of convergent evolution, having evolved independently so many times that researchers have largely stopped trying to maintain a definitive count. Venom, a toxic secretion delivered to another organism through a wound, has evolved independently in snakes, lizards, spiders, scorpions, jellyfish, cone snails, platypuses, slow lorises, shrews, and various fish species, including stonefish and stingrays. The number of independent origins is estimated conservatively at over 100 across the animal kingdom, with new examples continuing to emerge from genomic analysis of species previously considered non-venomous. What makes the venom case particularly interesting is the convergence at the molecular level within the broader convergence at the functional level. Many venoms from completely unrelated lineages contain the same classes of protein molecules: phospholipases, serine proteases, three-finger toxins. Not because these lineages inherited them from a venomous common ancestor, but because these protein families have been independently recruited into venom systems multiple times. The same protein types keep appearing in venoms because they have properties: stability outside cells, ability to disrupt specific physiological processes, resistance to degradation, that make them particularly amenable to venom function. The recruitment of existing proteins into new functions, evolutionary exaptation followed by specialization, appears to follow predictable pathways determined by the protein families that happen to be expressed in salivary and skin glands in the starting lineage. The adaptive landscape for venom molecules is constrained by the pool of proteins available for recruitment and by the molecular properties that venom function requires, and these constraints produce convergent outcomes across drastically different lineages. The saber-toothed morphology is a case study in what happens when the same ecological niche, taking large prey by throat or neck bites using elongated stabbing canines, is so persistently available over geological time that evolution fills it repeatedly as its occupants go extinct. True saber-toothed cats, the famous Smilodon and its relatives, belong to the subfamily Machairodontinae within the cat family. They evolved their elongated upper canines, reinforced neck muscles, and wide-opening jaw gape, independently of the feature's appearance in other lineages. But saber-toothed morphology has appeared in at least 6 different mammalian lineages across the Cenozoic era, including in a marsupial, Thylacosmilus from South America, that convergently evolved the entire suite of saber-toothed features, including blade-like upper canines, reduced lower canines, flange on the lower jaw to protect the upper canine when the mouth was closed, and reinforced skull and neck musculature. Thylacosmilus and Smilodon were separated by over 160 million years of separate evolution. Their last common ancestor was a small Mesozoic mammal with nothing resembling saber-toothed morphology. Yet they arrived at essentially the same anatomical solution, complete even to details like the lower jaw flange that would seem to be a fairly specific design choice. The saber-toothed apex predator niche is a peak on the adaptive landscape that evolution climbs toward reliably whenever a large prey ecosystem exists and a predator lineage is positioned to exploit it. The repeated loss of saber-toothed lineages, they don't exist today despite the long history of repeated evolution, suggests the morphology has a vulnerability, likely related to the mechanical challenge of using very elongated canines effectively in prey capture, that eventually disadvantages saber-toothed predators in competition with more versatile killing techniques as prey fauna change. The adaptive peak has a limit on how long it remains optimal as ecological conditions shift, and every lineage that reached it eventually fell off. Succulents in plants, the storage of water in thick, fleshy tissue, has evolved independently in desert-adapted plant lineages on different continents that never had the opportunity to exchange genetic material or ecological information. Cacti are the iconic succulent plants of the Americas. They store water in their swollen stems, have reduced or eliminated leaves to minimize water loss, conduct photosynthesis through their green stems, and protect themselves from herbivores and water thieves with spines evolved from modified leaves. This entire suite of adaptations to desert conditions is a spectacularly successful response to arid environments. Cacti are extraordinarily diverse in the Americas, occupying desert niches with impressive ecological success. African euphorbs, plants in the genus Euphorbia, look strikingly similar to cacti. They have swollen water-storing stems, reduced leaves, spines, and similar photosynthetic and physiological adaptations to arid conditions. They are not cacti and are not closely related to cacti. They belong to a completely different plant family that was geographically separated from cacti by an ocean and millions of years of independent evolution. Their spines are modified stipules rather than modified leaves, structurally different though functionally identical. They use slightly different biochemical pathways in their water conservation physiology. But the external appearance is close enough that non-specialists regularly confuse them. The convergence extends to the molecular level. Both lineages independently evolved the CAM photosynthesis pathway, a biochemical modification that allows carbon dioxide fixation at night rather than during the day, reducing water loss that would accompany daytime gas exchange. The same metabolic innovation, involving the same enzymatic machinery in a modified operational context, evolved in a range of unrelated plant lineages adapting to water-scarce environments across multiple continents. The physics and biochemistry of water conservation constrain the available solutions in the same way that the physics of hydrodynamics constrain the available body plans for fast aquatic predators. The repeated evolution of eusociality, the extreme cooperative social system where most individuals sacrifice personal reproduction to support the reproduction of a few, is perhaps the most behaviorally complex case of convergence in the biological record. Eusociality, with its defining features of cooperative brood care, reproductive division of labor, and overlapping generations contributing to colony function, has evolved independently at least 11 times in insects, multiple times in hymenopterans, ants, bees, and wasps, separately in termites, separately in thrips, separately in aphids, and separately in certain beetles and shrimp. It has also evolved in 2 mammals, naked mole rats and Damaraland mole rats, burrowing rodents in Africa that have independently evolved the same colony structure with a reproductive queen, and non-reproducing workers that characterizes social insect colonies. The conditions that favor eusociality appear to be specific enough. High genetic relatedness within groups, defensible resources worth collective protection, ecological conditions favoring philopatry, that when these conditions exist, the evolutionary pathway toward cooperative breeding and eventually eusociality opens up and tends to be traversed. The convergence is less about the specific mechanisms and more about the ecological and kin selection mathematics that make extreme cooperation evolutionarily stable once the initial conditions are in place. The naked mole rat case is particularly striking because mammalian life history and physiology are so different from insect life. History and physiology that the independent evolution of essentially the same social organization represents convergence across an extraordinary phenotypic distance. Everything about how naked mole rats are built is different from how a wasp colony is built. But the social structure, the queen, the workers, the cooperative defense and foraging, the suppression of reproduction in non-reproductive individuals through social and chemical means, is strikingly parallel. The limit of convergent evolution, the cases where similar selection pressures produce similar but clearly non-identical solutions, reveals the second layer of the phenomenon that pure examples of convergence sometimes obscure. Parallel evolution produces similarity that is intermediate in its degree. The same selection pressure operating on lineages with similar ancestral starting points produces similar but not identical outcomes. The repeated evolution of large body size in island isolation, the island gigantism seen in giant tortoises, Komodo dragons, and various bird species on islands lacking large mammalian predators produces similar outcomes across different lineages because the selection pressure and the ecological release are similar, but the specific anatomical outcomes reflect the different starting points. Developmental constraints further shape convergent outcomes by limiting which solutions are accessible from specific starting anatomies. A fish evolving toward terrestrial locomotion has a different starting toolkit than a tetrapod that has secondarily returned to water, and the convergent outcomes in both cases reflect both the adaptive optimum being approached and the developmental path available from the starting form. The wings of bats and birds converge in function, but diverge in structure because the available transformations of the forelimb differ between the two lineages' ancestral anatomies. These complications don't undermine the convergence principle. They enrich it. Evolution is not finding the same solution by identical routes. It is finding the same or similar solutions via different routes, constrained by both the destination, the adaptive landscape, and the starting point, the ancestral phenotype. The result is a biological world with strong recurring themes played in different keys, with variations that reflect evolutionary history, even as the melody reflects physical law. The philosophical implications of convergent evolution extend beyond evolutionary biology into questions about the predictability of evolution and the nature of biological possibility. If the same forms keep evolving independently, it suggests that the space of viable biological designs is not limitless, that evolution, despite its seemingly unbounded creativity, is repeatedly constrained to explore the same regions of biological space because those regions are where the adaptive peaks are. The implication is that evolution, given enough time and enough independent lineages, is somewhat predictable. Not in its details, not in the specific sequence of events that produces a specific organism, but in its broad directions. This has been tested experimentally. Richard Lenski's long-running E. coli evolution experiment, now running for tens of thousands of bacterial generations, has shown that parallel mutations appear in independent lineages under the same selection pressure. The same genes mutate in the same directions, producing the same adaptations in replicate. Populations that share only their ancestral starting genotype and their selection environment. The evolution of the same traits in independent lineages is not limited to geological timescales. It happens in real time, in controlled conditions, because the adaptive landscape is what it is. The question of whether life elsewhere in the universe, if it exists, would converge on forms recognizable to us, would independently discover the eye, would independently discover flight, would independently discover streamlined bodies for aquatic locomotion, is one of the most tantalizing extrapolations from the convergence principle. If the physical and chemical laws are universal, the adaptive landscapes they create should be universal too. The constraints that drive convergence on Earth should drive convergence anywhere life evolves under similar physical constraints. The professor's remark, "Evolution has a limited imagination," was partly a joke and partly the most compact possible statement of a deep truth. Evolution is not limited in the sense of being creatively deficient. The diversity of life on Earth is staggering, and the solutions evolution has found to biological challenges are frequently more elegant and more effective than anything human engineers have deliberately designed. But evolution operates within the bounds of what physical reality permits and what selection can reach from the starting point available, and those bounds turn out to create a landscape where the same solutions keep appearing. The dolphin and the ichthyosaur look the same because the ocean is the ocean, and water resistance is water resistance. And 150 million years of separate evolutionary history is no obstacle to arriving at the same streamlined answer when the question is the same. The eye keeps evolving because light is real, and spatial information encoded in returning light is genuinely useful, and nervous systems capable of extracting it survive better than nervous systems that can't. Venom keeps evolving because causing rapid physiological disruption in potential prey or predators is effective, and the molecular machinery available for recruitment into that function keeps being in the same places across animal lineages. Evolution found the streamlined body. Then it found it again. Then again, then 9 more times. And it will keep finding it for as long as fast aquatic predation is a viable ecological strategy and the physics of water remain what they are. That's not a limited imagination. That's the universe showing you what it considers a good idea.